So dear students, now I will talk about Unit 1. As already I shared this PPT and notes with you. So I will summarize in short the first part of Unit 1, that is various terminologies and sources of crude drugs and processing of crude drugs or herbal drugs. Okay. So this is the summary of this part of Unit 1. So more than 80% population in the world use directly or indirectly herbal medicines or natural medicine, or they use traditional system of medicine as per WHO. So what is traditional system of medicine? The medicinal system which is not main system of treatment or which is not allopathy is called traditional system of medicine, and the examples are in the context of India, Asia. The example is Ayush, Ayurveda, Yoga, or Unani, Siddha, homeopathy. So these are the systems which are for various years on the planet Earth. Ayurveda, for example, originated from India. Siddha system also originated from India, but its root was centered to South India, basically Tamil Nadu. Unani system of medicine started in Greece, which was called Yunan earlier. In modern it is called Greece, and from Greece through the influence of Hippocrates and other scholars, it went to the Iran, Iraq, and finally through Mughals it came into India. So this is the system of Unani medicine, and homeopathy system of medicine originated around 200 years ago in German, and the concept is like cures like. Likewise, Ayurveda, Siddha, Unani, they have their own principle of treatment, diagnosis, counseling, and other things. But yes, one thing common in all these systems is they all follow the treatment should be based on the root cause and not to treat the symptoms, like allopathy medicine, which is reverse in allopathy actually. And they all follow the they all follow the medicinal plants when it comes to treatment procedures. They make medicines through minerals, animals sometime, and also the medicinal plants. So now we move ahead with this basic background. So if you talk about drug discovery from natural products, animal or plants, basically that is medicinal plant. So the system starts, in this way. If I explain you, this is the, suppose for example we want to explore anti-cancer medicinal plant. So these are the various parts of the plant: fruit, leaf, flower, fruits, and other parts of the plant, root for example. So based on the part of the plant we are choosing, or based on the type of polarity, type of phytoconstituents, for example polar molecule, non-polar molecule, we will choose the battery of solvents. Then we will go for extraction of the plant material, then we will go for filtration, fractionation, and isolation. Once we get the suppose we assume that we got a pure chemical compound, we will go through various type of analytical procedures like NMR mass or spectroscopy or TLC like chromatography to confirm whether the molecule is pure or not. If it is pure, the next step is to confirm whether it is a novel molecule, that is first time it has been isolated from that plant, or first time in this earth it is being isolated first time, or it is a compound already available in other plant or in this plant. So what is dereplication? Dereplication means to confirm this is not a novel compound, it is already available in this plant or in other plant. After confirmation of the pure component, we will go for testing in the laboratory animals, in vitro testing, or finally clinical trial to confirm the drug is pure and safe and effective. Then through clinical trial, regulatory norms, it will become the drug. Allopathic drug it can be become. So in syllabus there are four words: herbal drug preparation, herbal medicine, and herbal medicinal product. So herb means anything obtained from plant and kept as such without much processing is called herb. Now for example, ginger is the basic example. You got ginger, you uprooted ginger from the ground, it will be herb. The moment you find some extract from the ginger, it will be called herbal drug preparation. Moving ahead, you made some capsule of the dried ginger powder, it will be called herbal medicine. And finally, if you go towards higher level of sophistication, if you are standardizing as a biomarker, and if the company is claiming each capsule or each gram of ginger powder contains 500 milligram of gingerol, it means it is standardized preparation, then it will be called herbal medicinal product, though all of them contain ginger. So if we get quinine from cinchona in pure fashion, it is approved by US FDA. It is having potent medicinal activity. It is single component and it has showing activity in human through clinical trial. And if there is well developed analytical method to quantify this chemical, then it is no more herbal. Yes, it is herbal from origin point of view, but from regulatory point of view, it is an allopathic medicine. Sources of the drugs. So we all know medicinal material are obtained from various part of the plant. For example, if you talk about cardioglycoside, digitalis, it is obtained from the leaves of digitalis purpurea family Scapholariaceae. Likewise, this is the list of all the herbal drugs in different parts of the plant as herbal drugs. We all know there are two types of drugs: unorganized drug, organized drug. Organized means if we see the portion or a tiny part of the plant material under microscope, and if we observe well defined cellular structure, it is called organized drug, like bark, root, stem, leaf. On the other hand, if we do the microscopy of oil, gums, mucilage, we cannot see crystal clear structure of the cells in the microscope. It means they are unorganized. Now selection, identification and authentication of the drug. Selection means to choose the medicinal plant on some logic is there, then to identify the given plant is so and so through various chemical tests like WHO, QC parameter is there. And finally, after identification, we have to authenticate the plant material by taking sign and by depositing a sample in the botany department of a college, because botanists are the qualified taxonomists to confirm the authenticity or the identity of the plant. We tell that herbals are safe. Yes, herbals or natural medicines or Ayurvedic medicines are safe. They are much safer compared to allopathic counterpart. But there are identity crisis. If we don't properly identify the plant material, they can lead to hazards. They can be detrimental for the user. These are the examples. So, for example, around a year ago, 10 kids fall ill after consuming castor seeds in Karnataka area, a province of India. So, yes, we should identify the plant properly before consuming those plants or the plant part. Why there is need? They all are Vinca rosea or Catharanthus roseus, the famous plant which gives world famous anti-cancer molecule which are indole alkaloids, and they are vincristine and vinblastine. They all are might be having same genus. They all are having genus Vinca, but they are different species. So if we want to explore the center white flower wala species, we cannot go for first and last one because they have different type of chemical. So if we compare allopathic medicine with allopathic medicines are they are not usually like polyherbal. They don't have too much chemical in single formula. For example, if we talk about Chanprash contain hundreds of chemical, so it is difficult to quantify and to set up a well-defined analytical procedure like allopathic medicine in case of polyherbal formulation. The best example to understand polyherbal formulation is Chanprash. It contain uh, 51, 50, 60 medicinal plants. Each plant contains around 100 medicament. So imagine how many total chemicals are there in Chanprash. How to do their NMR and mass. This is the method to identify medicinal plant, for example, DNA, DNA fingerprinting. For example, ashwagandha has various variety. So to confirm the authenticity or to differentiate two variety of ashwagandha, we can isolate the DNA of every plant, every species of ashwagandha. We can go for amplification of DNA through polymerase chain reaction. Then we can match the sequence with the authentic sample, and we can come to conclude, we can jump to conclusion, okay, this is the fingerprinting of authentic one and sample one, so both are having same type of DNA. These are the evaluation methods of crude drugs, taxonomical methods, herbarium, vouchers, herbarium just to identify the plant through botanist, macroscopical features, color, order, taste, size, shape, specific feature, texture, microscopy, physical chemical test, chromatographic methods, test for aflatoxin, test for molecular markers, and so on. As I told you, herbarium is to be deposited into the botany department to finally authenticate the sample. I mean the plant to be chosen for the research. Now the next topic is processing of raw material, herbal raw material. Processing means all the stages involved after collection to final shipping. To final shipping. So there are methods like primary processing and secondary processing. Primary processing involves basically garbling, means sorting or separation, washing to remove dirt, unwanted material. Blanching means to boil the material in the water to remove dirt. Then drying to remove moisture and to prevent the microbial infestation and to decrease the weight. Ultimately, it will lead to ease in logistics. Garbling to separate, washing means to remove dirt, unwanted material. These are the special treatment. For example, ginger is bleached using lime water to make it white in appearance, because we remove it from ground, it contains dirt and of white color is there. To make it shiny white, we have to treat with ginger. It is not the condition, but this is the usual commercial practice. So to remove stock from the clove is also called garbling. Blanching, as I bleaching is to remove the color using bleaching powder. Washing to remove dirt. This is the video. You can see on YouTube also how these machines are used to washing the material. Then blanching, I told you. Now drying is important. Primary processing, it involves removal of moisture to remove the microbes also, by removing the moisture. These are the various ways of drying: hot air oven, likewise. Then we have sun drying, then artificial drying, shade drying. Then these are the fresh leaf. If we dry in shade, this is the color. If excessive temperature, it will different color is there. Cutting again to increase surface area, to do the faster processing, faster handling and removing moisture. Then secondary processing involves aging or sweating. Sweating means to keep drug for some time to increase the potency. For example, cascara salgada. Baking roasting, to heat in water. Fumigation, baking roasting means to bake to remove the unwanted things. Fumigation means to remove the germs by fumigation under sulfur dioxide. Sometimes there is irradiation to by cobalt 60 to remove the germs before shipping to US. For example, mangoes are shipped to US by treating them through ionization radiation. Packaging is important before we go ahead. The packaging means to give proper shape, to make product sellable, to increase shelf life by retaining the active moiety, active chemical in the herbal preparation. They are the type of packaging materials you can see in there. Gunny bags are there, tin container, plastic pouches, polythene bags, and so on. Then we should keep drug in amber water to prevent the effect of light exposure. Then these are the example of some germs present, microbes present in the crude drugs, which are to be removed or to a certain level to increase the shelf life of the drug. Thanks, that's all for the first part of the syllabus of unit 1, that is the definition of 4 herbs and those definition, then processing, and that's all for today. In next video, I will talk about second part of the unit. So I am Dr. Amit Gangwal, Associate Professor at prestigious SVKM NMIMS Global University, Dhule. Thank you.